Hello students, welcome to EPG Pathshala. I am Suma Saad from Jamia Hamdard University, New Delhi, and the topic is Biopharmaceutical Classification and Biowaivers, and paper is Product Development Part 2. First of all, we are talking about learning objects. In this module, we will study about what is BCS and how drugs are classified under BCS. Other than that, parameters of BCS such as solubility, permeability and dissolution. Other than that, application of BCS with particular reference to BCS based biowaivers and other application of BCS classification. Now we will talk about introduction. Pharmacological therapy is essential in many disease treatment, and it is important that the medicine policy is intended to offering safe and effective treatment with affordable price to the population. In generic drug development, lot of money as well as time is equipped in conducting bioequivalent studies involving human participant. One way to achieve this through biowaivers defined as the replacement of in vivo bioequivalence study by in vitro studies. Under biowaivers, based on solubility, permeability and dissolution data, a generic drug can be introduced into market. Applicability of biowaivers is based on BCS classification proposed by Amadon and co-workers in 1995, which allowed the classification of drug based on solubility and intestinal permeability parameters in 4 classes. The core idea is that in vitro methodology centrally embracing permeability and solubility with qualifications related to the pH and dissolution may qualify drug products for a waiver of in vivo bioequivalence study. The regulatory bodies of US, that is Food and Drug Administration, and of Brazil, that is Anvisa, use the biopharmaceutical classification system as mean to allow biowaiver or immediate release solid oral dosage form containing drugs with high solubility and permeability, that is class 1 drug. The European Union agency, that is EMA, responsible to indicate candidates to biowaiver has considered eligible the class 1 of BCL. Additionally, drugs which have high solubility and low permeability, that is class 3rd, which they show every rapid dissolution 85% in 15 minutes. The World Health Organization further extends the applicability of biowaivers for drugs in class 1 and class 3rd, and also drugs poorly soluble and highly permeable class 2nd when they have characteristics of weak acid and high solubility in pH 6.8 beside rapid or very rapid dissolving. Thus, in short, the objective of the BCS is the predict in vivo performance of drug products from in vitro measurement of permeability, solubility and dissolution. In this method, we will study classification of drug based on BCS, parameter based on which drugs are classified, the application of BCS to get biowaivers for the drugs. Now, classification of the BCS. The BCS classification and its use in drug delivery. The BCS characterizes drugs into 4 classes according to their FDA solubility and permeability. The FDA promulgated the BCS system as a science-based approach to allow waivers of in vivo bioavailability and bioequivalence testing of immediate release solid oral dose from the class 1 high solubility, high permeability drug, which such drug products also exhibited rapid dissolution. The waiver is based on a triple trial rationale, where point A is high solubility ensure that drug solubility will not limit dissolution and thus absorption. Point 2, high permeability ensure that drug is completely absorbed during the limited transit time through the small intestine. And the last one is the rapid dissolution ensure that the gastric emptying process is the rate limiting step for absorption of highly soluble and highly permeable drug. Purpose of BCS guideline: expand the regulatory application of the BCS and recommend methods for classifying drug. Point 2, explains where a waiver for in vivo bioavailability and bioequivalence study may be requested based on the approach of BCS. Now, the goals of BCS guideline. First goal is to improve the efficacy of drug development and the review process by recommending a strategy for identifying expandable clinical bioequivalence tests. CNS. Point 2, to recommend a class of immediate release solid oral dose for which bioequivalence may be assessed based on in vitro dissolution test. Point 3, to recommend methods for classification according to dosage form, dissolution along with the solubility, permeability characteristic of drug substance. Now the BCS categories and its 4 class and within examples. First class, BCS class 1, which have high solubility and high permeability, and the examples are chloroquine, metoprolol, paracetamol, propranolol. Class 2, which have low solubility and high permeability, examples are ketoconazole, danazole, carabinazole, etc. Class 3rd, high solubility and low permeability, and the examples are atenolol, cimetidine, captopril, ranitidine. Now, class 4 drugs which have low solubility and low permeability. Examples are cyclosporin A, furosemide, allergic acid, Taxol. Now, class 1 drug which have high solubility and high permeability. Those compounds are well absorbed are under in this category. Point 2, their absorption rate is usually higher than excretion, and the point 3rd, this class drugs exhibit high absorption number and high dissolution number. The class 1 drug are not those in which either solubility or permeability is limited within the target regions of the GI tract. The drug release in such cases can be modulated using controlled release technology. Class 2nd, high permeability and low solubility. This class relates to the case in which solubility or dissolution rate is limiting and thus significantly affect absorption and BA. The technologies under this class include the approaches such as classical micronization, stabilization of high energy state, use of surfactants, emulsions or microemulsion system, solid dispersion and use of complexing agent such as cyclotaxins. The technologies under this class include soft gel such as nanoemulsion, nanosuspension, nanocrystals are treated as hopeful means of increasing solubility and BA of poorly water soluble active ingredients. Point 1, those compounds have high absorption number and low dissolution number. Point 2, in vivo drug dissolution is then a rate limiting step for absorption except at a very high dose number. Point 3, these drugs exhibit variable bioavailability and need the enhancement in dissolution for increasing the bioavailability. Point 4, these compounds are soluble for design the SR and CR formulation. In vitro, in vivo correlation is usually expected for class 2nd drugs. Now we are talking about class 3rd, low permeability and high solubility. The absorption is limited by the permeation rate, but the drug is solvated very fast. Drug permeability is the rate limiting step for drug absorption, but the drug is solved very quickly. These drugs exhibit a high variation in the rate extent of drug absorption. Since the dissolution is rapid, the variation is attributable to alteration of physiology and membrane permeability rather than the dose form factors. Manipulating the site or rate of exposure or perhaps by incorporating functional agent into the dosage form to modify the metabolic activity of enzyme system are included in class 3rd technology. The technology under this class including oral vaccine system, gastric retention system, high frequency capsule and telemetric capsules. Now class 4 drugs which have low permeability and low solubility. Those compounds have a very poor bioavailability. Usually they are not well absorbed over the intestinal mucosa and a high variability is expected. The drugs of this class are prolembitic for effective oral administration. Fortunately, extreme example or class 4 compound are the exception rather than the rule, and these are rarely developed and marketed, but a number of examples of class 4 drugs do exist. For example, cyclosporin A, furosemide, ritonavir or Taxol. Now we are talking about the parameter of BCS. In this heading, we will study the 3 basic parameters of BCS. Number 1, solubility, number 2, permeability, and number 3, dissolution. We will also study about how these parameters are evaluated. The first parameter to discuss is solubility. Solubility is the amount of a substance that has passed into solution when equilibrium is attained between the solution and excess substance at a given temperature and pressure. According to BCS, a drug is said to be highly soluble if its highest dose strength is soluble in 250 ml or less of aqueous media over the pH range of 1 to 7.5 as per FDA guideline or 1.2 to 6.8 as per WHO guidelines. The volume estimate of 250 ml is derived from the typical volume of water consumed during the oral administration of a dosage form. This boundary value is a reflection of the minimum fluid volume anticipated in the stomach at the test drug substance should be determined at the 37 plus minus 1 degree centigrade. A sufficient number of samples should be evaluated to accurately define the pH solubility profile. A minimum of 3 replicate solubility determination in each pH condition should be carried. Traditionally, the equilibrium solubility at a given pH and temperature is determined by the shake flask method. According excess drug is added in solvent in which solubility is to be determined, medium and shaking at the predetermined time, usually 24 hours or longer. The solution is then filtered and analysis of filtrate is carried to determine the amount of drug present per ml of solvent. Method other than that, the traditional shake flask method such as acid or base titration method can also be used with justification to support the ability of such method to predict equilibrium solubility of the test drug substance. The next parameter to discuss is permeability. The permeability is a dynamic and complex process that involves the permeation of the drug across biological membranes. The arrival of the drug into the bloodstream is made by the absorption pharmacokinetics process. The transport mechanism include passive diffusion through the enterocytes and the junction between the enterocytes as well as active mechanism employing energy and carrier. A drug is considered highly permeable when the extent of absorption in human is 85% or more, based on mass balance determination or in comparison with an intravenous dose. On the other hand, according to FDA, one drug is classified as highly permeable when the fraction of the absorption dose or the absolute bioavailability is equal or greater than 90%. This criterion can be considered conservative because there are many reports of drugs that are generally considered well or completely absorbed that present fraction of absorbed dose less than 90%. This suggests that a threshold rating of 85% may be appropriate in definition of high permeability. The need to obtain data relating to intestinal permeability of drug has raised the development of various models for the determination of permeability. The models may be in vitro based on cell monolayers. The cell monolayers such as CoCA2 cells and TC7 clone mimic in vivo intestinal epithelium in humans. Second, animal tissue base. Out of animal tissue based model, everted rat sac technique is widely employed for determination of permeation of drug. Point 3, in situ method. In situ perfusion of intestinal segment of radons is frequently used to study the permeability and absorption kinetics of drugs. The biggest advantage of the in situ system is the presence of an intact blood and nerve system in the experimental animals. In situ experiments involve perfusion of drug solution prepared in physiological buffer through isolated cannulated intestinal segment. Absorption is assessed based on the disappearance of drug from the intestinal lumen. Fourth, in silico. Computational screening has received much attention in the last few years. In silico model can accurately predict the membrane permeability of test drug based on lipophilicity, H bonding capacity, molecular size, polar surface area and quantum properties. The next parameter to discuss is dissolution. The dissolution can be defined in a narrow sense as the process by which a solid substance enters the solvent to form a solution. However, in the broad sense of the word, it is more than simply measuring the rate of solubility and can be more correctly described as a physical assay to predict the release of substance for a given area in quantity and adequate time. The absorption of a drug contained in the solid dosage form after oral administration depend on its release, solubilization and dissolution under physiological condition and posterior permeability through the gastrointestinal tract. The first two steps present a critical nature and therefore in vitro dissolution may be relevant to predict the in vivo performance. An immediate release product is considered rapidly dissolving when no less than 85% of the labeled amount of the drug substance dissolving within 15 minutes using USP dissolution apparatus. First at 100 rpm or apparatus second at the 50 rpm in the volume of 900 ml or less in the following media: 0.1 NaCl or simulated gastric fluid or pH 4.8 buffer and pH 6.8 buffer or simulated intestinal fluid. One way to evaluate the dissolution is by determining the dissolution profile, graph of percentage of drug dissolved versus time, which is a relatively quick and inexpensive way to evaluate solid dosage form and allows obtaining kinetic parameters. Among several methods investigated for dissolution profile comparison, F2 is the simplest. Moore and Flanner proposed a model independent mathematical approach to compare the dissolution profile using two factors, F1 and F2. Two important factors to compare the drug products are similarity factor and differentiation factor. Similarity factors, as the name specifies, it stresses on the comparison of closeness of two comparative formulation. Generally, similarity factor in the range of 50 to 100 is as acceptable according to US FDA. It can be computed using this formula as you can see on the screen, where N is the number of dissolution sample times, RT and TT are the individual or mean percent dissolved at each time point, T for the reference and the test dissolution profile respectively. The similarity factor should be between 0 and 100. It is 100 when two comparative groups of reference and the tests are identical and approaches 0 as the dissimilarity increases. Difference factors. Difference factors focus on the difference in percent dissolving between reference and the test at various time interval. It can be mathematical computed by using you can see on your screen. Therefore, the factor directly compare the difference between percent drug dissolved per unit time for a test and reference product. Now, we will discuss about the application of BCS. BCS is widely used in design and development of innovation drugs, new drugs forms, in clinical pharmacology and also by regulation agency of several countries as the scientific approach for testing of waivers on bioavailability. Given below the application of BCS in different fields. Now, we will discuss about BCS biowaivers. The first and foremost application of BCS is BCS based biowaivers. The term biowaiver is applied to a regulatory drug approval process when the dossier is approved based on evidence of equivalence other than through in vivo equivalence testing. Biowaivers means to obtain waiver of for carrying out expensive and time consuming BA and BE studies. A biowaiver has been regarded as an official approval of the waiver for conducting a bioequivalence study in the context of an application for drug approval process. The BCS based biowaivers applying during pre-IND, NDA and ANDA and post-approval phases. BCS based biowaivers are acceptable for immediate release solid oral dosage formulation containing one or more of the APIs mentioned above if the required data ensure the similarity of the submitted pharmaceutical product and the appropriate pharmaceutically equivalent comparator product. BCS based biowaiver has been an important and cost saving tool in approval of generic drug. Now, the criteria of BCS based biowaiver. Biowaivers are based on the biopharmaceutical classification of the active ingredient. Currently, BCS class 1 and some class 3rd compound are eligible for biowaivers. Point 1. The drug substance should be highly soluble and highly permeable. Point 2. An IR drug product should be rapidly dissolving. Point 3. The drug should not be narrow therapeutic index drug. Point 3. Excipient used in the dosage form should have been used previously in FDA approved IR solid dosage form. Point 5. For waivers of an in vivo relative bioavailability study, dissolution should be greater than 85% in 30 minutes in the 3 recombinant dissolution media, that is acid media such as 0.1 NaCl or simulated gastric fluid USP without enzymes, a pH 4.5 buffer and pH 6.8 buffer or simulated intestinal fluid USP without enzyme. And the last one is for waivers of in vivo bioequivalence tests and reference products should exhibit similar dissolution profiles under the dissolution test condition defined for rapidly dissolution products. Two dissolution profile may be considered similar when compared using the F2 matrix when both the test and the reference products dissolve 85% or more of the label amount in less than 15 minutes in all 3 dissolution media recommended above. A profile comparison is unnecessary. Now, the other application of BCS is NDA, that is new drug application, and ANDA, abbreviated new drug application. The principle of the BCS classification system can be applied to NDA and ANDA approval as well as to scale up the post approval changes in drug manufacturing. This has resulted in the following outcomes. IND and NDAs. BCS based biowaivers are applicable to the formulation intended to be marketed where changes in composition, component of the formulation or method of preparation occur to the clinical trial formulation as long as the formulation has rapid and similar in vitro dissolution profile. This approach is usually if the drug is highly soluble and highly permeable, BCS class 1 drugs, and both the pre and post change formulation are pharmaceutically equivalent. ANDA. For highly soluble and highly permeable drug substance formulated whose dissolution is rapid as defined in section 3rd US FDA guidelines, and in vivo bioequivalence study can be waived provided that the reference listed drug product is also highly soluble and both the test product and the reference product have similar dissolution profiles. That is, this approach is suitable only when the test and the reference products are pharmaceutically equivalent. Where feasible, the choice of dissolution apparatus, that is USP 1 or 2nd, it should be limited to that stabilized for the reference product. Post approval changes. BCS based biowaivers can be requested for significant post approval changes, that is level 3 changes in components and composition to a rapidly dissolution immediate release product intended for oral use. Further, the drug substance should be highly soluble and highly permeable provided that the dissolution remain rapid for pre and post change product and both pre and post change product exhibit similar dissolution profile. Approval of generic. Because generic were required to meet essential safety, efficacy and BE criteria, few were approved under these regulations till 1984. In 1984, the Drug Price Competition and Patent Term Restoration Act was passed and established the abbreviated New Drug Application Procedure, that is ANDA, permitted the FDA to approve generic product for drugs that had already been found safe and effective and formulated the criteria for pharmaceutical equivalence and bioequivalence. Provision for post-approval changes for BCS class 1 rapid release oral dosage form has made it possible for generic pharmaceutical companies to obtain approval without necessarily conducting their bioequivalent study. The next application of BCS is that it is used as framework for optimization of a new chemical entity. Last application is BCS for pharmacological screening. Pharmaceutical drug discovery and delivery group are using human drug absorption studies for understanding the biopharmaceutical properties of early drug candidates. The HDA, that is human drug absorption, provides significant guidance to a pharmaceutical formulation scientist in selecting a route of experimentation, clinical development and improvement of bioadhesive system if the drug is absorbed from the selective area of the intestine. According to Linsky, a rule of 5 is widely absorbed for screening of compounds that are likely to have poor absorption profile. According to this rule, the poor absorption or permeation is more likely when there are more than 5 H-bond donors and the molecular weight of the drug moiety is more than 500 and the log P is over percentage, and the last one is there are more than 10 H bond acceptors. Compounds that are substrate for the biological transporters are an exceptions to the rules. The last and conclusion part of this module is based on the BCS in regulatory practice. Throughout the past decade, the BCS has become an increasingly important tool in drug product regulation worldwide. By presenting a new paradigm in bioequivalence is the critical step that connects the physical drug product with the clinical properties claimed on its label, ensuring continuing quality of the innovative products and the generic products. By relieving the fundamental parameter dictating the in vivo oral drug absorption process, the BCS is able to ensure BE by mechanistic tools rather than empirical observation. If two drug products that contain the same active pharmaceutical ingredient have similar GI concentration time profile under all luminar condition, then a similar rate and extent of absorption is ensured for three products, that is, they are bioequivalent. Thus, BE can be guaranteed based on in vitro dissolution tests that provide the mechanistic proof for similar bioavailability rather than empirical in vivo human study. This is the regulatory waiver of in vivo BE based on the scientific and mechanistic rationale provided by the BCS. Initially, waiver of in vivo BE were accepted only for scale-up and post-approval changes, but later the Biowaiver principle was extended to the approval of new generic drug product, thus avoiding unnecessary human experiment and reduce cost and time of developing generic IR oral drug products. The solubility classification of a given drug is based on the highest dose strength in an IR product. According to current FDA guideline, drug substance is considered highly soluble if the highest strength is soluble in 250 ml or less of aqueous media throughout the pH range of 1.2 to 6.8. The volume of 250 ml is derived from typical BE study protocol that prescribe administration of a drug product to fasting human volunteer with a glass of water. Otherwise, the drug substance is considered poorly soluble. A drug substance is considered highly permeable if the extent of intestinal absorption is determined to be 90% or higher. Otherwise, the drug substance is considered poorly permeable. The permeability classification is based either directly or extent of intestinal absorption of a drug substance in humans determined by mass balance or in comparison to an intravenous reference dose, or indirectly on the measurement of the rate of mass transfer across the human intestinal membrane. Alternatively, animal or in vitro models that predict human intestinal absorption, that is, intestinal rat perfusion model or epithelial cell culture model, can be used. An IR product is characterized as rapid dissolution if not less than 85% of the lipid drug amount is dissolved within 30 minutes using USP apparatus 1 at 100 rpm or using apparatus 2 at 50 rpm in the volume of 900 ml or less of each of the following media. First, acid media such as USB simulated gastric fluid without enzyme. 2. pH 4.5 buffer, and third is pH 6.8 buffer or USP simulated intestinal fluid without enzyme. Otherwise, the drug product is considered to be slow dissolving. Up to now, the FDA has implemented the BCS system to allow waivers of in vivo, BA and BE testing of IR solid dosage form for class 1 high solubility and high permeability. As for class 3 high solubility and low permeability drugs, as long as the drug product does not contain agents and or excipient that may modify intestinal membrane permeability. In vitro dissolution tests can ensure BE. The absorption of a class 3 drug is likely limited by its permeability, less dependent upon its formulation, and its bioavailability may be determined by its in vivo permeability pattern. If the in vitro dissolution of a class 3 drug product is rapid under all physiological pH condition, its in bio behavior will essentially be similar to oral solution, and as long as the drug product does not contain permeability modifying agent in vitro dissolution test can ensure BE. Hence, biowaivers for BCS class 3 drugs are scientifically justified and have been recommended. Thank you.